Our next speaker is Matt Spitzer. He completed his training in immunology at Stanford with Gary Nolan and Edgar Engelman, developing experimental and analytical methods to model the state of the immune system using high-dimensional single-cell data. He then moved to UCSF in the summer of 2016 as a Parker Fellow and a Sandler Faculty Fellow, and he's now an assistant professor and an investigator of the Parker Institute for Cancer Immunotherapy, and his lab focuses on systems-level coordination and regulation of immune responses, in particular in the context of cancer immunology and immunotherapy. So I will pass it over to you, Matt.
Terrific. Thank you so much, Stacy. And thank you for organizing such a great event today. It's really fun to have a chance to connect all the dots between the groups that collectively make up the Institute. And there's so many fantastic collaborations and intersections that we're already underway and looking forward to. So thanks so much to both Alex and Stacy for putting this together. I'm very happy to tell you a bit about our work today. Generally, in my group, we focus on understanding how the immune system makes decisions. This is sort of the key question that keeps us up at night. And generally, we think about this as a multi-tiered or multi-scale type of process. So we think a lot about what's happening at the organismal level, either in patients or in mouse models that we use quite frequently. We think about how the immune system regulates choices that are made at the level of the physiology of the organism. And so, as Stacy said, a lot of the work that we do is in the context of cancer immunology. And we can think about this in a fairly simple way, which is to say that the immune system can either be tolerant to a cancer and enable it to grow, or the immune system can actively fight against a cancer to reject it. And so what are the components and the molecular and cellular elements that help to make that choice? We think a lot about how the immune system is compartmentalized across tissues. So the immune system is relatively unique in terms of organ systems in the body, in as much as it's a distributed system across space. So immune cells constitutively infiltrate essentially every tissue across the organism. And somehow these cells that are distributed across really large distances from each other still need to communicate to coordinate their behavior to make coherent choices or collective choices when the physiology of the organism changes, say in the context of an infection or the development of a cancer. And we also think a lot about the cellular level of diversity. The immune system is made up of lots of different types of cells, and they all have unique roles and responsibilities. And again, somehow these cells need to communicate to coordinate their behavior. We also think a lot about how the immune system is a decentralized system. So there's not a master regulator cell that provides instructions to every other type of cell, but somehow these cells collectively make decisions through exchanging various signals and cell-cell interactions. And one of the key processes that we work a lot on in my lab is trying to understand the molecular interplay that takes place when T cells get activated. So Jimmy's already told you quite a bit about T cells. T cells are critical for controlling many types of disease processes across the body. And the primary cell that activates or primes a T cell is called a dendritic cell. That's of the myeloid cell lineage. And dendritic cells provide three key signals when they activate T cells. The first, they present foreign antigens in the context of MHC molecules. And T cells with a T cell receptor specific to that peptide MHC complex will sense the presence of it. Dendritic cells also provide co-stimulatory signals. So these are additional stimulating signals that allow a T cell to get activated. The prototypical co-stimulatory receptor interaction would be CD80 and CD86 on dendritic cells binding to CD28 on T cells. And dendritic cells also secrete cytokines that help to activate and polarize T cells, IL-12 being an important one. And when a T cell sees these three signals, it gets activated, it starts to clonally proliferate, expands, and it differentiates from a naive cell that's never experienced its foreign antigen before into a diverse suite of antigen experienced cells that have different roles and responsibilities. And we can sort of simplify this diversity by thinking about effector cells that are very good at killing. They carry out lots of effector functions, but they're relatively short-lived. Or memory cells, which, as their name suggests, persist for long periods of time and help to control secondary challenges. So in the case of an infection, whether you re-expose to the same antigen or the same pathogen later on in life, or in the context of cancer, these cells can promote the control of cancer recurrence. And these cells, once they're primed, generally in lymphoid tissue, which is the spleen and draining lymph nodes across the body, these cells will then leave these lymphoid tissues. They'll migrate through the circulation, and they'll infiltrate into the target tissue, wherever the disease cells are, where they undergo their effector function. And here you'll see, generally during a productive immune response, a large number of effector cells carrying out their effector function, and a smaller number of memory cells that persist in this target tissue and turn into what we call resident memory T cells. Now, in the context of cancer, things go a little bit awry. So T cells experience chronic antigen stimulation because tumors persist for long periods of time. And so a key process that was described a number of years ago by Rafi Ahmed's group, including John Wary, who's really pioneered this field, is the concept of T cell exhaustion. So when T cells continuously see their foreign antigen, instead of differentiating down the trajectories that I've told you about, the sort of conventional effector and memory trajectories, T cells instead differentiate toward an exhausted lineage where they lose their effector functions gradually over time. And you'll note in the schematic that one key receptor that these cells upregulate is PD-1, which many of you know is a key target for cancer immunotherapies. And so this concept a while back led to the idea that anti-PD-1 as a therapeutic was perhaps reactivating these exhausted cells that persist in the tumor microenvironment. We did some experiments using single cell approaches, and I won't go into the real intricate detail of the single cell analysis for this particular piece of background data, but I'll show you, more or less how we came to this conclusion. But we came to the understanding that when immunotherapies are effective, immune cells are actually recruited from outside of the tumor. So sort of challenging this idea that immune cells within the tumor were primarily responsible for being reactivated and mediating the effect of immunotherapy. And so our single cell analysis pointed at this, but as we like to do in the lab, we then designed a causal experiment to try to prove to ourselves as best we could that these immune cells from outside of the tumor were in fact important. And so what we did was to take mice that have cancers that generally respond to checkpoint inhibitors, and you can see that represented in the blue curve, which are mice treated with anti-PD-1 antibodies, as compared to the green curve, mice that are left untreated. And you can see that the tumor size is controlled well by anti-PD-1 in this mouse model. And what we did was to also treat mice with another molecule called FTY720 that prevents immune cells from leaving the lymph nodes and the spleen. So these lymphoid tissues outside of the tumor. And essentially, we asked the question, are the immune cells already in the tumor sufficient to mediate the response to immunotherapy? Or do you need the participation of these new immune cells that come in from outside of the tumor to mediate this effect? And what we saw was pretty clear cut that if you block the trafficking of immune cells from outside of the tumor, in fact, you lose the efficacy of the immunotherapy. So that's the red curve here. And you can see that the tumors in these mice grow very similarly to if the mouse was untreated altogether. So this really suggested to us that systemic immunity, what's happening outside of the tumor, is required for effective cancer immunotherapy. And now we've known for a number of decades that tumors alter immune cell function within the tumor microenvironment. And T cell exhaustion is a good example of that process, but this extends beyond T cells to a number of other immune cell types in the tumor microenvironment as well. What we felt was missing from this picture was trying to understand how tumors might impact the immune system outside of the tumor. Now we knew that these cells outside of the tumor were critical for immunotherapy. So how does tumor development alter the systemic immune state? And I'll start by telling you a project that we worked on in this area and just published last year. And then I'll go on to tell you some unpublished data from future directions that this work has led us down. So what we started to do to try to understand this question of what's happening to the immune system outside of the tumor was to analyze a variety of different mouse models of cancer that have different types of immune infiltrates within the tumor. So what I'm showing you here is the composition of immune cells within 8 different types of cancer in mouse models. This represents breast cancer, melanoma, pancreatic cancer, colon cancer, and glioblastoma. So a wide variety of different mouse tumor models. And you can see that each model creates its own unique tumor microenvironment. So the composition of immune cells within that tumor is distinct. And another way to show this is by principal component analysis. So we're reducing the dimensionality of our data here into a two-dimensional plot and saying, what are the major drivers of variance? And you can see that each tumor model occupies a unique space in this chart. So each tumor is creating its own unique immune cell environment locally within the tumor. What we didn't necessarily expect, but was equally striking, is that each tumor was also driving equally important and large changes to the immune system outside of the tumor. So what I'm showing you here is the same principal component analysis, but now of tissues outside of the tumor across all these models. In gray is the state of the immune system in a healthy mouse that doesn't have cancer, and each color represents a different kind of cancer. And so what you can see is that each cancer is driving a distinct macro environment systemically across the organism, changing the types of immune cells that are present across these various tissues. And that to us was very striking because it suggested that there's a lot of biology that we really haven't been paying very close attention to that's happening outside of the tumor microenvironment, but systemically to the immune state. Now, I told you that one of the key cell types that we focus on in the lab are T cells. And so we wanted to understand whether this process of T cell exhaustion was happening locally in the tumor or if it was happening systemically across the organism. And so we used single cell analysis here to identify 29 different unbiased subsets or clusters of T cells across all of the tissues in these mice. And we did this as a time course study. So you're looking from left to right over chronological time as the tumors grow and expand, C standing for control mice that don't have tumors, and 35 representing large, bulky tumors that are at the ethical endpoint. So these animals have essentially succumbed to their disease at this time point. And what you can see is across all of these tissues, there are dramatic changes in the composition of T cells. And this is summarized at the far right in the stacked bar plot, where each color represents the deviation in each specific tissue. So each tissue is color-coded, as you can see at the top here. So you can see that there's a lot of dynamic change taking place across each tissue in terms of the composition of the T cells. But something that was striking to us was that the exhausted CD8 T cells, which really belong here in cluster 10, these are uniquely expanding in the tumor, and we really didn't see these expanding across all of these other sites in the organism. And we confirmed that by using a different definition of exhausted T cells that Andrea Scheidinger's group at Memorial Sloan Kettering has defined using these different markers, CD101 and CD38. You can see that these cells don't exist in the spleen, but they do become abundant in the tumor as tumors progress. So this suggested to us that there are a lot of changes happening to the T cell compartment, but they're not the same outside of the tumor as they are inside of the tumor. So that then led us to ask the question, does the immune system respond normally outside of the tumor to a threat that's completely distinct from the cancer? Does cancer change the choices that the immune system makes across the body? And this has become actually quite relevant now in the age of COVID, because we've seen that cancer patients tend to have worse clinical outcomes in COVID infection or SARS-CoV-2 infection compared to healthy individuals. And so we asked this question in our mouse models by taking mice with and without breast cancer and by challenging them to make responses to pathogens, bacteria and viruses that drive strong T cell responses typically in a healthy mouse. And we asked, does the T cell response happen normally in this context? And what we found is no. So this was a mouse model of breast cancer. And as you can see in the control mice, these mice mount a very robust CD8 T cell response to Listeria, which is an intracellular bacterium. So Ki67 is a marker of cell proliferation, and you can see robust CD8 T cell proliferation. Granzyme B is an important protein involved in effector function and killing, and you can see robust upregulation of granzyme B in these cells. But when we looked in the mice with cancer, this is AT3 is a mouse model of breast cancer here. You can see that both the proliferation and the effector function of these T cells was markedly diminished. And this is just summarized here at the right, where the gray bars represent control mice and the red bars represent mice with cancer infected with either a virus, LCMV, or this bacterium, Listeria monocytogenes. And you can see in both contexts, mice with cancer mount really dysfunctional responses. Now, this is also important because these pathogens don't have antigens that are shared with the tumor. And so the context of T cell exhaustion requires chronic antigen stimulation to drive dysfunction of T cells. Whereas here, we're challenging to make a response to an antigen that's completely unrelated to the cancer. So again, this really suggested to us that this process was distinct from exhaustion. And one other way to answer this question is we know that exhaustion is permanent. It's not a reversible process. There are distinct epigenetic changes that take place to prevent those cells from becoming reactivated. And so we asked the question, are these cells from outside of the tumor, these T cells that are not mounting good responses to the pathogen, are they also permanently dysfunctional, or are they only temporarily dysfunctional based on the context of the cancer that it's creating in the body? And so we took T cells from control mice or mice with cancer, and we labeled them with two different dyes, and then we transferred them into a control mouse at the time of infection. So we're asking here, does the origin of the T cells matter, or does the context that the T cells try to get activated matter? And what you can see is the cells proliferate, they'll dilute out this dye, so they'll move from right to left in the plot. And you can see an equal number of cell divisions between the T cells that originated from a mouse with cancer or a mouse without cancer. So this really suggested that the T cell deficiency that we saw in cancer is not T cell intrinsic, but is really the context of the physiological context of cancer that it creates in the body. And so as I told you, dendritic cells are key cells for activating and priming T cell responses. And so we then wanted to ask whether if T cells themselves were not permanently impaired, perhaps dendritic cells upstream of T cells were impaired in this context of cancer. And that is indeed what we saw. So when we looked at an earlier time point, we saw that dendritic cells were failing to upregulate these key co-stimulatory proteins and activation markers that drive T cell proliferation. We then asked if we could rescue this response by stimulating the dendritic cells. And in fact, that was the case. So we know that this molecule CD40 is an important key co-stimulatory molecule on dendritic cells. And if we activate CD40, we can increase the expression of CD86, which helps activate T cells. So we asked, can CD40 rescue this T cell response in the context of a tumor-bearing mouse? And we found that indeed it could. So if we took these mice with cancer, we infected them with Listeria, and we also added an agonist antibody to activate the CD40 pathway in dendritic cells, we were able to restore the magnitude of the CD8 T cell response to near close to what we would see in a healthy animal. And this was notable because checkpoint inhibitors were actually unable to reverse this process systemically in the body, suggesting that really acting on dendritic cells might be a critical place to reverse this systemic dysfunction that takes place in the context of cancer. Now, one of the things that we had seen is this process isn't permanent, it's reversible. And so we also wanted to know if we resected the tumors surgically, if we just remove the tumor burden, could we restore the responsiveness of the animal? And in fact, that's what we saw. So if we surgically remove the primary tumor, we're actually able to restore the magnitude of the CD8 T cell response, in this case to infection. And then lastly, of course, we wanted to understand what the molecular drivers were of these systemic immune alterations. And we found that the tumor cells are chronically producing a few key inflammatory cytokines, IL-1 and GCSF. And we found that if we blocked these molecules using neutralizing antibodies, that we were able to prevent a lot of the systemic immune dysfunction that takes place in the context of cancer. So here on the plot, we're looking at changes in the composition of immune cells outside of the tumor. And you can see that by neutralizing IL-1 or GCSF, we prevent many of the changes that take place in the context of cancer. And this is true specifically in the T cells as well, but IL-1, not GCSF, is actually the primary driver of these alterations in T cells. And so to summarize this first part of the talk, hopefully what I've shown you is that tumor development induces an immune macro environment outside of the tumor as well, and that new immune responses are impaired in the context of cancer. The silver lining here is that these processes are reversible. They're still plastic. They can be manipulated. Activating dendritic cells outside of the tumor is a good way to do this. And IL-1 is a central mediator of some of these dysfunctional changes that we see in the context of cancer. And so I want to tell you now about some work that's unpublished, that's sort of taking a step back and asking some fundamental questions about dendritic cell T cell interactions and how productive T cell priming works. What we realized when we were doing these studies in the context of cancer is there were some critical gaps in our understanding of how good T cell priming takes place. And so what we did was to design a very fundamental experiment to really understand in a lot of cellular and molecular detail the longitudinal trajectories that take place across immune cells during an acute infection when a strong T cell response is mounted. And so here we're still using our model of Listeria as a good way to drive type 1 immune responses. And each day over the course of the infection, we collected the spleen, which is the site of infection, as well as the blood to understand what circulating factors were being produced over time. And something central that we saw is that the response to this infection is highly dynamic. So what we're looking at here is a UMAP plot, and Jimmy's already beautifully introduced the concept of these dimensionality reduction plots, so I won't go over that again. But what you can see is that using our single cell analysis approach, we can really, in a lot of molecular detail and cellular detail, see all of the different types of cells that are present across the organism. And if you now look at where cells are sitting in these different activation and phenotypic spaces over the trajectory of an immune response, you can see really dynamic changes each day over the course of an infection. And this is summarized down below in the principal component plot, where you can see this trajectory that moves from the bottom right corner of an uninfected mouse through a phase of acute innate immune cell activation, represented by the blue and pink dots on the left, through a trajectory of adaptive immune cell activation that represents the purple dots as we move higher through infection space over time. So at each time point, we really see critical changes taking place in the immune response. And the real question that we were asking was, when do we see T cell priming taking place? And what we found is that these type 1 dendritic cells that are really critical for priming CD8 T cell responses are actually only highly activated at 1 day during infection, which is day 2 of the infection. You can see that the activation of these cells really goes back to normal essentially by day 3. So there's a very narrow transient window in which dendritic cells are being activated during infection. And you can see that here. Now we're looking at the expression of a lot of key activation markers on dendritic cells. And you can see that these are very highly upregulated on day 2 post-infection, but immediately return back to baseline for most of these proteins by day 3. Now, this activation trajectory in the dendritic cell is highly organized. So what we've done here is to reconstruct a pseudo-time trajectory of dendritic cell activation. And as you can see, there's a very linear trajectory through time of how these cells are becoming activated, and a very coordinated molecular program that they undergo to upregulate a lot of these key co-stimulatory proteins at a very specific moment in time, and then to shut them back down, reversing in this trajectory as they become deactivated. Now, of course, we wanted to know what are some of the key drivers of this very acute window of dendritic cell activation. And we found that at this same point in time, on day 2 post-infection, there's a wave of interferon gamma that's secreted by bystander T cells and NK cells. And so we saw that on day 2, using now a reporter mouse for interferon gamma, we see a peak of cells that are expressing interferon gamma at this time point. And we also see that dendritic cells at day 2 are signaling through the STAT1 pathway, which is activated by interferon gamma signaling. So at the top here, you can see that these DCs on day 2 have upregulated phosphostat1. And importantly, this does not happen in vitro when we co-culture dendritic cells alone with Listeria. So this is not a direct sensing response, but a response to the interferon that's provided by these lymphocytes. Now, as I've told you, the majority of this interferon is produced by NK cells and by T cells. And we found that if we specifically deplete T cells, we actually are able to abrogate this wave of interferon production. And we're also able to prevent this activation window of dendritic cells. So here in the blue are cells that are coming from a normal infected mouse. And you can see the expression of many of these key proteins is reduced if we deplete T cells or if we block interferon gamma itself. So this very transient window of interferon gamma production is activating this narrow window of dendritic cell activation in the animal. Now, we thought this was really interesting, this idea that dendritic cells are only highly activated for a short period of time, because antigen-specific T cells before the state of an infection will be randomly distributed across the organism. And so what we did was to construct a mathematical model to understand what % of these antigen-specific cells will recirculate to the site of the infection cumulatively over time. will all of the antigen-specific cells arrive by day 2, which is really this key window when dendritic cells are being activated? And what we found is really no, a relatively small fraction of T cells are likely to come back and encounter the activated dendritic cell on day 2. And we experimentally wanted to ask whether this was true. And so we turned back to our friend FTY720 that allows us to inhibit T cell trafficking. And we saw that if we inhibit T cell trafficking, we actually reduce the proportion of T cells that are becoming activated, turning on the expression of NIR77, which is a high fidelity indicator of recent signaling through the T cell receptor. So new cells are trafficking and arriving at the site of infection, even at past this critical day 2 time point. And this led us to then ask what happens to a T cell if it sees a dendritic cell at this peak activation time point, or if it happens to arrive just after this dendritic cell activation window. And so we adaptively transferred OT1 T cells, which are antigen specific to the listeria, on each day over the course of an infection. And what we found is that the differentiation trajectories of these T cells were markedly changed if they happen to encounter a dendritic cell before or after day 2. So what we're looking at here is the expression of 2 key proteins that delineate effector versus memory T cell differentiation. KLRG1 leads cells to differentiate into effector cells. CD62 ligand leads to the differentiation of memory cells. And what you can see is that T cells that encounter dendritic cells before day 2, a healthy proportion of these cells get primed and differentiate into short-lived effector cells that turn on the expression of KLRG1. But if a T cell instead encounters a dendritic cell after day 2, on day 3 or day 4, all of these T cells instead differentiate into central memory cells, long-lived memory cells. And we thought this was really an important understanding because tuning the balance between effector cells and memory cells is a key component of an adaptive immune response. We need to make enough short-lived effector cells to engage in effector function, such as killing and cytokine production, but we also need to make a sufficient number of memory cells to protect over the long term against reemergence of a tumor or reinfection by the same pathogen. And so what this suggested to us is really that this narrow window of dendritic cell activation is an important way to tune this balance between how many effector cells you make and how many memory cells you make. And just to reinforce this point, now we're looking in a high dimensional view at the differentiation of these cells if they're transferred at day 0, so they encounter dendritic cells at peak activation, or at day 3 where they miss this peak activation. And you can see that in dimensionality reduced space, these cells really occupy distinct areas. And this is defined by the expression and upregulation of an effector versus memory proteins. Now, the last question we wanted to ask is whether dendritic cells themselves were sufficient to lead to this result. You could imagine, I've shown you that over the course of the infection, a lot of things are changing in the immune system. And so is this really just the dendritic cell activation state, or are there other key components perhaps that are influencing T cell differentiation? And so we isolated dendritic cells from either day 1.5 or 2.5 infected mice. This gives us a few hours to create the cultures before they reach day 2 or day 3. And we added those cells then to naive CD8 T cells that had never experienced antigen before. And what we saw is that we could, in fact, in vitro, recapitulate this change in T cell differentiation that we were seeing in vivo. So dendritic cells that are at their peak activation state here give rise to more short-lived effector cells in vitro that lack expression of CD62 ligand, whereas dendritic cells that just miss this peak activation window give rise to largely memory cells in vitro as well. So yes, the activation state of the dendritic cell is sufficient to drive this change in T cell differentiation trajectories. And so what I've shown you here is that during an acute infection, there are rapid changes that take place in immune cell states. One of the key things that changes here is the activation state of dendritic cells that undergo a very transient window of activation at day 2 post-infection. This is driven by interferon gamma signaling. And in turn, this tunes the balance between effector cell and memory cell differentiation of T cells. So a T cell that sees a dendritic cell at peak activation will largely differentiate into a short-lived effector cell. And a T cell that sees a dendritic cell, even shortly after this peak window of activation, will almost exclusively differentiate into a memory CD8 T cell. And of course, we're really interested now in understanding exactly which components of these responses are altered in the context of T cell activation in cancer and a variety of other contexts where T cell dysfunction is a critical component of pathology. And so with that, I will wrap up. I'd really like to acknowledge a couple of fantastic grad students in the lab who led these projects, Bree Allen and Kamir Hayam Galvez. Kamir is now a postdoc in the SatPathy lab, so he's stayed within our family of the Gladstone-UCSF Genomic Immunology Institute, as well as all of the other members of the lab who have contributed to these and many other projects ongoing, and some key collaborators, and of course our funding sources. And with that, we'll wrap up here and happy to take any questions.
Thanks, Matt. Great talk. There are actually a bunch of questions in the Q&A. We're running a little bit behind, so I'm only going to ask you one of them, and then you can go in and type answers to the others when you have a chance. But Lisa Butterfield would like to know, how do you envision that these carefully orchestrated TDC crosstalk events happen in human cancer settings, and can we intervene or optimize these interactions?
Yeah, that's a really great question, Lisa. So what we think is probably happening in the context of cancer is that this peak window of dendritic cell activation just may never exist due to lack of ligands or pattern recognition receptors that are important for driving this activation, as well as this wave of interferon production. And so what we think is probably happening is that we're getting more of this sort of tonic memory cell differentiation without the production of a really good burst of short-lived effector cells, and that then through chronic antigen stimulation, these cells eventually become dysfunctional when they arrive in the tumor. And so something we're really thinking about is, can we reproduce this transient activation window in vivo to drive really strong effector responses by priming new T cells, not previously engaged T cells, in the context of cancer? And of course, that's a long ongoing effort and something that you've been thinking carefully about too. So I'm looking forward to carrying on that discussion offline.